Hello ladies and gentlemen and welcome back to C.A.R. Entertainment. I'm your host Don Hamilton, and today guys we're going to be talking about epigenetics as a part of our A22 biology revision videos guys. For further ado, let's get straight into it, because epigenetics, you're probably wondering what the hell is that? Which you might be, you might not be, because considering you're probably watching this, you're probably studying A22 biology and therefore you probably should know a little bit about it, but in case you don't, this is what it is. Well, epigenetics is the study of changes in gene expression that do not involve changes in the base sequence of DNA, but instead they involve changes in gene expression that are caused by the changes in the cell environment. It involves some genes being switched on and others being switched off. When the gene becomes more active or it becomes shut down, the genes will be permanently or temporarily called epigenetic modifications. Okay, so a first note you may need to know is that the epigenetic modification will not change the DNA sequence itself. As we know, it will be caused by a change. It will be a change in gene expression. And note 2 is that a mutation will alter the DNA sequence. So an epigenetic modification will not, but a mutation will. So the first method of epigenetic modification is DNA methylation, and this is whenever methyl groups will be added to a base of DNA with a chemical tag that is altered throughout its life in a response to biochemical signals. So what you essentially really need to know is that the methylation of DNA will prevent the attachment of RNA polymerase, which will inhibit the process of transcription in protein synthesis. Methylation will prevent the gene therefore being expressed, and the gene will be silenced, and therefore the protein that that gene will code for will not be made. The details of DNA methylation and its process then. Well, once methyl is added to cytosine, where the G is followed by the C, so in your base sequence you have CGA for example, your methyl group will be added to that cytosine because it was GCA, I should say, because the C is followed by the. I was right the first time, CG, where the C is followed by a G. So whenever you have a C base followed by a G base, so G's after that C base, immediately after, and methyl will be added to that cytosine, and this will be called the CPG sequence. These CPG sequences will then be grouped in CPG islands. So all these CPG sequences in the gene that have a methyl added to it will be grouped in CPG islands. When a critical number of these CPG islands is methylated, the DNA will be switched off and transcription will not take place. So when DNA is methylated, it will switch at the start of the gene, and the DNA methylation will also be long-term and stable. This explains why proteins are associated with liver function are only produced in the liver and nowhere else. Other parts of the body will have genes involved for coding for liver proteins, but these will be switched off. So obviously, in the stomach you might have a protein that is coded for, but it might be a liver protein, and therefore it'll be switched off through the process of DNA methylation. The epigenetic modification will have DNA methylation, where it will pass from cell to cell as mitosis takes place. The second method is histone modification, because as we know, DNA is wound around histones which produce chromatin, but whenever the DNA histone complex is tightly condensed, the DNA will not be transcribed, and this is because histones are altered by adding acetyl, methyl, or a phosphate group, which will affect whether the DNA is condensed or not. So for example, if you add an acetyl group, then the DNA will be less tight, and therefore it's easier for RNA polymerase to bind. But if you remove this acetyl, then the DNA will be wound more tightly, and therefore transcription will be suppressed. Histone modification is relatively short term, but it is important, as if you increase or decrease the expression levels of particular genes. The significance of epigenetics is that they occur during embryonic development, where cells will differentiate, and this will ensure some genes are switched on and some are switched off. If you go back to immunity in A21, this is where this sort of comes in terms of making sure you don't go through self-immunity harm. Age-related diseases are associated with epigenetic changes. Epigenetics can cause tumor suppressor genes to be silenced or oncogenes to be activated, and drugs can treat cancers by reversing epigenetic changes. Epigenetic modifications are usually long-term and inheritable, which are passed on during mitotic division from the parent to the offspring. Thanks for watching this video. I've been Don Hamilton from C.A.R. Entertainment, and I'll see you next time.